Good morning. It's April 17th, and this is your daily brief in science. Here's everything you need to know. OpenAI unveils GPT Rosalind, a biology-focused AI model optimized for biology, drug discovery, and translational medicine to assist workflows across chemistry, protein engineering, and genomics. The model is available as a research preview in ChatGPT, Codex, and the API for qualified customers through a trusted access deployment, with a free life sciences research plugin for Codex, linking scientists to more than 50 tools and data sources. Collaboration is already underway with Amgen, Moderna, the Allen Institute, Thermo Fisher Scientific, and others to apply GPT Rosalind in research and discovery. Experts caution that while these tools boost productivity, frontier AI models remain imperfect and may struggle with basic tasks in some domains, underscoring ongoing limits. Market context shows Alphabet stock sentiment skewing toward a strong buy, with potential upside around 15% based on current targets. A Codex-based PubMed evidence constellation pulls PubMed results, classifies papers by evidence type, and visualizes them by year, delivering a mobile-friendly, self-contained HTML/JS fragment. The core thesis argues that domain-specific architecture matters as much as domain-specific prompting for high-stakes science, with production volume the key test for HelixGen in the coming year. Beta partners report substantial productivity gains, cutting literature review timelines from about 3 weeks to under 3 days, signaling faster early target identification in drug discovery. Atlas and Constellation demos showed roughly 18 minutes total, automating large portions of data synthesis and visualization that previously took hours. Historically, AI-discovered drugs have struggled to reach clinical trials, with none reaching Phase 3 to date. The rollout comes amid biosecurity concerns around AI-trained biological data, prompting calls for tighter controls on training data. Industry context notes AI-driven shifts in pharma, with collaborations like Eli Lilly and In Silico Medicine, and broader moves in discovery, diagnostics, and manufacturing optimization. A new intranasal therapy from Tiziana is drawing attention as the only fully human anti-CD3 monoclonal antibody in clinical exploration, with potential implications for neurodegenerative and neuroinflammatory diseases, including Alzheimer's and multiple sclerosis. Tiziana reports ongoing clinical activity in NASPMS, noting that 14 patients in the Expanded Access Program showed improvement or stability within 6 months. CEO Ivor El-Rafi highlights broader potential for Foralumab across MS, Alzheimer's, and ALS, and points to favorable safety signals from earlier intranasal Foralumab data in non-active secondary progressive MS. TLSA's stock trades around $1.34, up about 66% over the past year, signaling investor interest. The stock rose roughly 5 to 6% intraday on the news, as markets weighed the mechanistic relevance and possible future development against the limited, non-peer-reviewed data. The full preprint is available on BioRaxiv and has not undergone peer review yet. The Form 6K includes a press release, and the BioRaxiv preprint detailing the long COVID study, which remains unreviewed. Analysts caution that TLSA is a clinical-stage biotech with encouraging preclinical signals but no revenue or earnings yet, advising readers to consider risk alongside potential. Investors are advised to review TLSA's stock page and screening tools for context on early-stage opportunities and risks. The report ties preclinical data, early clinical signals, ongoing trials, and recent financing into Tiziana's broader neurology-focused strategy, with experts noting a broad therapeutic window for intranasal Foralumab and activity in both early and late stages after infection without harming antiviral immunity. Tiziana also highlights prior safety signals and clinical responses in expanded access for NASPMS and the ongoing Phase 2A trial, NASPMS, NCT 06292923. A new study in Nature Astronomy reveals jets from the Cygnus X1 black hole moving at about half the speed of light and carrying roughly 10% of the total energy from matter accreting onto the black hole. For the first time, researchers measured instantaneous jet power around 10,000 solar luminosities and speeds near 355 million miles per hour, or about 540 million kilometers per hour. The team ties jet activity to the black hole's feeding rate and growth, offering insight into how jets interact with their environment and possibly shape galaxy evolution. Using nearly 2 decades of data from the VLBA and EVN, scientists reconstructed time-resolved, high-resolution images that show a dancing motion of the jets caused by interactions with the companion star's stellar wind. Jet measurements were achieved by tracking their wobble in combined radio telescope images, yielding a clearer picture of jet structure than previous efforts. Cygnus X1's history stretches back to its discovery in the 1960s, with ongoing gas accretion from its companion-fueling jet activity. The analysis draws on 18 years of high-resolution radio imaging from a global telescope network and uses jet bending from the stellar wind, plus computer modeling, to estimate jet power. Energy estimates come from multi-telescope radio imagery, showing the jet-stellar wind interaction bends the jets and makes them appear to dance, enabling power calculations. Jets are conceived as being pushed in opposite directions by the stellar wind, a dynamic that provides the basis for determining their power through bending and modeling. Researchers including Steve Prabhu and James Miller-Jones presented the findings, framing them as a major step in understanding one of the universe's most energetic phenomena. A new study from University College Dublin's Centre for Bio-Nano Interactions, published in Nature Materials, shows that certain nanoparticles entering a cell acquire a condensate corona made from the cell's own proteins and RNA, effectively delivering a small biological program. These corona nanoparticle complexes act as active interfaces that carry functional biomolecules and can unlock endogenous cellular gateways, enabling delivery to intracellular environments that are normally hard to reach. The researchers outline a design pathway for nanoparticle prototypes able to target specific cellular compartments, integrating biomolecular science with nanoengineering and biomedical research. Framed as a blueprint for sending therapeutically effective biological messages to hard-to-reach locations, the findings point toward treatments that could reverse certain intractable diseases rather than merely manage them. The study notes a link between misdirection of this intercellular messaging system and processes like tumor metastasis, underscoring relevance to oncology research and potential cancer therapies. Researchers used embedded magnets to capture the corona-containing droplets in transit, enabling readouts of how messages transfer between cells while preserving their integrity. Magnet-assisted capture showed that the droplets relay intact messages from source to destination, with the corona detaching inside recipient cells and helping cargo escape degradation. The messaging droplets, trapped during transit by embedded magnets, reveal how the coating detaches to facilitate cargo release and escape from the cell's degradation system before final delivery. Transferred biomolecules remain active after delivery and can directly influence the function of target cells. Proteins and RNA carried by the corona remain functional in recipient cells, suggesting practical potential for delivering medicines to previously inaccessible body regions. Overall, the discovery uncovers natural cellular gateways and a robust delivery mechanism that could transform biomedical interventions by leveraging the body's own messaging infrastructure. The system can transport cargo such as proteins and RNA across biological barriers with high efficiency and immune-evasive properties, preserving messages during transit and targeting recipient cells. A new study uncovers a bidirectional brain-fat body axis in fruit flies that links immune signals to learned avoidance of a pathogen's odor. Activated receptors trigger octopamine release in neurons, which stimulates dopamine production in fat cells, and that dopamine travels to the brain to boost learning-related activity tied to avoiding the odor associated with pathogenic bacteria. The result is a shift from initial attraction to the odor of harmful bacteria to avoidance after infection, revealing a conditioned taste aversion as a survival mechanism. The work was a collaboration among the University of Bonn and Leipzig, Tohoku University, and University Hospital Bonn, with funding from the German Research Foundation and related networks, and published in Neuron with open access. Participating institutions include Bonn, Bonn University Hospital, Leipzig, and Tohoku University in Japan, supported by the DFG, iBehave, and the Human Frontier Science Program Organization. The paper, Wei Wang and colleagues, A Bidirectional Brain Fat Body Axis for Pathogen Avoidance, appeared in Neuron on April 16, 2026, with Digital Object Identifier 10.1016/J.neuron.2026.03.026. The study integrates interactions among brain, immune system, and adipose tissue, and suggests similar mechanisms could occur in humans due to adipose tissue's ability to produce neurotransmitters that influence brain function and appetite. There is translational potential for humans, as mammalian adipose tissue also secretes neuroactive compounds, indicating conserved brain fat communication that could affect pathogen avoidance, Cygnus behavior, and possibly eating disorders. The authors propose this mechanism may exist in mammals and humans, with implications for understanding the metabolism-immune-brain axis. Future work aims to dissect how adipocyte dopamine synthesis and transport to the brain are regulated, identify receptors and downstream pathways, and understand how nutritional status modulates the brain-fat axis in pathogen defense. Using a model organism, the study clarifies brain-body interactions that promote survival by avoiding contaminated food after an immune challenge, with findings published in Neuron. Overall, the research reframes fat tissue as an active neuroendocrine organ capable of influencing cognitive processes and behavior through immune-metabolic signaling. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.